This conference will now be recorded.
Just a minute.
Yeah, yeah.
Uh, Yesterday voice was not clear, so I am introducing again. We'll start with beginning. Go-
Okay.
Good evening to all. Today is the fourth session in Beshaja Rachana Parishad. For today's session, we have an eminent speaker and pharmacologist, Dr. Urmila Aswar, madam. Currently she is an associate professor working with Pune College of Pharmacy, Bharati Vidyapeeth, Deemed University, Pune. She has completed her MPharm PhD in Pharmaceutical Sciences with specialization in pharmacology from the Department of Pharmaceutical Sciences, Nagpur, and PhD from Pune College of Pharmacy. She has 19 years of academic and research experience in total. Uh, Previously, she has worked with the National Institute of Pharmaceutical Education and Research, NIPER, Guwahati, and Siaghad Institute of Pharmacy, Pune, also with Indus Biotech, Pune. She has guided about 25 MPharm students and currently guiding 3 PhD students. She has received PC Dandiya Award for 2 times, OD Gulati Award, UNAS Award, and Achari Award from Indian Pharmacological Society, and recently Best Oral Presentation for her PhD student by Nipicon 2022. These are the names uh, awards given annually at the Indian Pharmacological Society. She has received Best Research Project of the Year 2010-11 by 54th IPC Trust IPA Pune branch. She is the recipient of Best Teacher of the Year award twice in 2011 and 2015 from Siaghad Institute of Pharmacy. She has published about 35 research publications in various peer-reviewed international journals. She has made more than 60 national and international presentations. She has completed 40 preclinical industrial projects. She is a reviewer of various national and international peer-reviewed journals, also works as an associate editor for some journals. She is involved in creating cognizance of various preclinical techniques by arranging hands-on training for pharmacology students. She is also associated with Lila Poonawalla Foundation, Pune, for promoting girls for higher education. Personally, we have worked in some research project related to Ayurveda. Uh, with this brief introduction, I request Madam to please start her session. So, Madam, please start. Thank
Yeah.
you.
Thank you, sir. Thank you, Chinmay sir, for giving this opportunity. And we have worked together for some of the Ayurvedic formulation where we wanted to develop them as validate them in preclinical studies, and we could get some good results. And in future also, we are collaborating for certain formulations where we feel that this is going to work in uh, maybe taken up as a drug. So as yesterday I spoke that when we compare Ayurveda and traditional Chinese medicine, because both are they are very old traditional medicines which are used by respective countries. So we see that the progress is made a lot in traditional Chinese medicine as compared to Ayurvedic medicine. So the reason could be the strict regulation which we have in India, or maybe uh, mm, government bodies, they are not that much cooperative, though we can see that with the establishment of Ayush, we can see that now it is happening. And so let us hope that in future we'll be getting most of the Ayurvedic formulation as drugs. Okay, so let me start with my presentation. From bench to bedside, new drug development process. So every drug which is used by the patient, it has to cross a long journey where it is studied for its efficacy as well as toxicology before it comes to the patient hand. So that we have already these slides, some of these slides we have already seen, and where I have given the molecules, the APIs, um, which is converted into the tablets. So a drug, as all the medicines, we already I told you that the drugs, these are the medicines for internal or external use by human being or animal, and the substances which are intended to be used in diagnosis, treatment, mitigation, or prevention of any disease or disorder in human being or animals, including preparations which are applied on human body for the purpose of repelling insect like mosquitoes. So this is the definition which is given by DNC Act of India 1940. So a drug is any chemical or biological substance. It can be from synthetic or non-synthetic origin. We have seen that previously, as I said, there are certain figures which we must appreciate, and the time taken for a new drug development, it was 7.9 years. It was 7.9 years in 1960, and now it is almost 12.8 years in 1990. So we are seeing that the drug development process is becoming very long in recent years, and this is happening because the strict regulations which are coming up by the government, and as well as in the clinical trial where the patients are involved, so patients are also understanding their own rights. So that's why we can see that the changes in the consent procedures, then changes, the amendments which are coming up in the clinical trials. So schedule Y, we can see the amendment has happened in 2001, and that talks about more about that the experiment should be conducted in a very ethical manner, and documentation should be very proper, and every experiment should be in the favor of the patient. So that's why we can see that the number of years are increasing in the drug development. Now let us understand why these new drugs are needed. I mean, there is no reason why we should ask this question, because we have recently seen the COVID pandemic, and we understood that in future also these type of pandemics or the newer diseases may come, and we should be prepared with the drugs. So yes, there is an unmet medical need. There are new diseases which are coming up, infectious diseases, Alzheimer's, obesity, AIDS, and the prevalence also keeps on changing. Where when we were thinking that India is coming out of infectious disease, and then we saw that India is more suffering from the diseases like diabetes, then cardiac problems, diabetes, Alzheimer's, and this is happening because of the change in the lifestyle, as well as the increase in the normal aging. So that's why we can see that Alzheimer, Parkinson's are also coming up as newer diseases, or we can say more people are suffering from it. So these are some reasons. Other than that, we have certain drugs still available, for example antipsychotics, but they do suffer from certain side effects, and that's why we require to develop new drugs in this area. Then downstream health costs like Alzheimer's, spinal cord injury, still we do not have very good drugs in this area, and that also affects overall, affect the because it's a chronic disorder and health cost is high. So we require that the medicines which will be cost effective. Then cost of therapy like interleukins. Now we can see that the interleukins, then TNF alpha inhibitors are coming up as newer drugs. Monoclonal antibodies are also coming up as newer drugs, but they cost very high, and that's why we require the drugs which will be of less cost. Then cost to the individual country, such as depression. So there are certain diseases which has become very much highly prevalent in certain countries, especially in developed country. So we can see depression is one of that disease, and therefore it cost high to the country, because overall, if the young population undergo these type of diseases, then it overall affects the working of that country, and it affects the growth of the country. So this example we can see from Japanese population, where we can see lot of youngsters as well as old people are suffering from depression. Sustained industrial activity. So as a part of sustained industrial activity, we require the new drugs to be discovered, because this is a continuous R&D procedure. The pharmaceutical industry, they employ thousands of people, and they make a massive contribution. Therefore they want that the new drugs should come into the market, so that there will be continuous, or there will be continuous flow um, of the money in the industry and into the market. So other than that, the patent expiry keeps on happening for new for drugs, and off patent drugs are also considered, that the people make a derivative of it. industries, they make derivative, or they use it in a different form. So that also becomes one opportunity for drug development. Let us understand now the initiatives which are there in drug discovery. So the initiatives are like choosing the disease, choosing the drug target, identifying a bioassay, finding out a lead compound, isolation and purification of the lead compound, structural determination of the compound, likewise. And similarly, the drugs which are coming up, they come from different sources such as organic synthesis, molecular modification, isolation from plants, or genetic engineering. So there are huge number, like we are not only in drug discovery we are becoming strong, but we are also becoming strong in organic chemistry, we are also becoming strong in analysis, we are also becoming strong in isolation, characterization. So yes, we these all fields are helping us to find out the newer drugs, newer targets, and that's why the process can be faster. That is what we have seen during COVID drug development. So let us see one by one these initiative. So choosing the disease. Now for our industry, so drug development will be something like industry or institute, because in Western countries we can see that lot of drug discovery happens at institute also, and many examples are there with where the drug for example, Viagra has been it has come from the institutional research. So choosing the disease, so it is to concentrate on developing drugs for diseases which are prevalent in developed country. So this is very underlined sentence that every country has their own diseases, and they should keep drug development prioritizing that for that disease. And we see that for India also, that India a lot of drug development happens for infectious disease. Other than that, to consider the economic factors, a huge investment has to be made towards the research R&D of the drug. So we have to consider economic factors also. Then the research is carried out on elements such as migraine, depression, ulcer, obesity, flu, cancer, diabetes, cardiovascular disease. So all these diseases, the prevalence rate is very high, and we see that the lot of research is being done in this area, whereas less research is carried out on the tropical diseases of the developed world. For example, the conditions like malaria. So it all depends upon the prevalence of the disease, how much a country want to invest in that particular disease treatment, and the overall market, how much how the drug will be commercialized. So all these factors are responsible for drug development. Let us go next. Choosing the drug target. Now second is choosing the drug target. A drug target can be a receptor, an enzyme, or nucleic acid. It can be certain biomolecules also. So understanding them is very very important. Understanding where this drug is going to act, how the drug is going to act, is very very important. So this allow whether the agonist or antagonist is required to be designed. Now with the help of now the docking studies and combinatorial chemistry studies, we are able to identify the receptors. We are able to see the binding of for the molecules with those receptors, and then going for synthesis. So this type of softwares, Schrodinger software, or certain good softwares which are available in this area, help us to do the molecular docking, to identify the target and drug interaction, and to develop the drugs. So this is the choosing the drug target again becomes a important initiative from medicinal chemistry point of view. So this allows, so in this case, for example, this is one example which I have given here, that desipramine, which is an antidepressant, and it inhibits the uptake of noradrenaline from the nerve synapses, and thereby it improves depression. This is an old theory which was given previously, but now we have understood that along with noradrenaline, serotonin is also important. So with the research in this area, in the central nervous system, in the field of depression, we are understanding more and more. We have understood that depression is not only affected by noradrenaline, but it is also due to the lack of serotonin. And now again, if we go much in detail in these studies, we understand that serotonin are of 14 receptors are available for serotonin, and all these 14 receptor has not still been identified. Few of them have been identified. Few of them, their role has been identified in depression, but some of these receptors are still required to be studied. So this type of yeah, the availability of resources, and the huge amount of literature which is getting available across the globe help us to develop new drugs. So in this case, when we understood that serotonin is important neurotransmitter, fluoxetine was developed as antidepressant, and it is one of the best-selling antidepressant drug. But considering the different pathophysiological conditions involved in depression, we feel that still the better drugs are required to come in this area. Discovering the drug target. So now this is again because of the advancement in the research area, we are able to identify the certain chemical substances which were initially unknown. Okay, so this is why, that is why certain receptor like PPRL, PPAR gamma, or maybe the mTOR type of receptors, so these receptors we are identifying, and we can feel that there are, these receptor might be involved in certain type of pharmacological effects or physiological effects, and they can be targeted. So the challenge is to find out the chemical that will interact with this receptor, and they will act as a suitable drug targets. So this is one important thing which is coming up. Then we should also talk about specificity and selectivity of the drugs. Specificity and selectivity is very, very important. The more the selective a drug is there for its target, the less chance that it will interact with other target, and it will have undesirable side effect. So this is very, very important, that a drug should be very specific and selective in its action. For example, penicillin. So penicillin inhibits the cell wall synthesis only of the bacteria and not of mammalian cell. So this is the selectivity and specificity which can, where the drug can identify between two species. So this type of effect should
Hello,
be.
just madam, just to interrupt, are you changing
Yes,
slides or it is the same slide?
I have changed, sir. Can we? This is, I have changed, discovering target specificity and selectivity. I'm on. Can you see the change in slides?
Yeah, message is coming. Slides are not changing. Is it the same slide?
That's
Can
not there.
someone please? Can someone please check? Mayuri, can you
It's
see
not
the
there.
slide?
Yes, the slides are not changed. It's
Yes,
second
that's
number
like
slide
Yes,
till
ma'am,
now.
it is not changing.
It is not changing. Is it now changing?
Now it is.
Okay, so maybe I have to put it not in the F5 mode. Okay. So just hold on. I'm just doing it properly again. Is it now correct? Now we can see choosing the disease. Can we see?
Yes, yes,
Yes, ma'am.
yes.
Can we? Yeah.
Yes, ma'am.
Full screen. Is it full screen now? Full screen, no?
No,
Yes,
ma'am.
ma'am.
No.
Choosing the drug target. Now, is it going? Target specificity and selectivity.
No, ma'am. It's previous slide.
Okay, so
Choosing the drug disease.
okay. So what I will do is I will just use, I will not use the whole um, complete mode. Okay. Switching the disease we have done. Now can you see the change in the slide, sir? Choosing the drug target.
Uh,
Yes.
yes, ma'am.
Okay, fine. So I will
Yes,
keep like this.
ma'am. Yes.
Yeah. Okay. Thank you. So we have seen that the drug should be very much selective when it is acting, and it should not affect the normal cell of the body, and it should affect only the desired cells. For example, in case of penicillin, so it should affect the cell wall of the bacteria, and should not affect the human cell. Okay, so we also require drug which will be which not be only specific against the species, but which also be a target specific. It should be target specific and selective within the body. So we have so many drugs available. They act on different receptors, and so for example we have the drugs which are beta 1 and beta 2 receptor agonist. So we want that the drug should be specific to beta 2 receptor, or the drug should be very much specific to beta 1 receptor. So these type of, again, in within the receptor also, within the body, the different receptors are present, and we want the drug should act on one type of receptor. So this is very important, and also targeting the drug to specific organ and tissue. For example, we want that the beta 1 agonist drugs should act on beta 1 receptor which are present in the heart. So this type of specific effect and selective effect is very much desired whenever we are talking about the drugs, new drugs, so that the side effects should be reduced, undesirable effects should be reduced. But sometimes it is not possible, because many of the diseases are multifactorial. That means they arise due to changes in uh, different enzyme system, ion channels, or receptors, expression of certain receptors. So, for example, I would like to give here is hypertension, where we see that hypertension is not only happening because of one reason, it could be because of many reasons, such as overactivation of beta adenoreceptor. Then it could be because of overactivation of renin angiotensin system. So here we require the drugs which will a good drug will be something which will act on many of these. So that is the pitfall of if we make the drug very much specific. So with the example I have given here again is for the asthma. Second example I'm giving for asthma. So in asthma we find that one drug treatment doesn't work, so we require to give a combination beta 2 agonist. One anti-inflammatory drug has to be added with it. Corticosteroid is added normally with beta 2 agonist. So these type of combination are required to treat the disease. In hypertension also we see that vasodilators combined with the angiotensin inhibitors or beta 1 blockers. So these type of combinations are normally seen in chronic diseases. So that is the disadvantage, that if we become more specific, then there are certain diseases where we have to take care. Now, other than that, choosing the target, choosing the disease, the very important thing is choosing the bioassay. So, a new drug discovery will always happen with certain tests. There will be in vitro test and there will be in vivo test. In vitro means when you are using the outside the biological system, you are using petri plate or you are using test tube, and you are seeing certain type of enzyme inhibition in this plate or in the test tube. Whereas in vivo studies means when these drugs are tested in biological system in the body. So so these two tests are very very important, and developing these tests, now we see that lot of tests are already available. A lot of research paper they talk about these tests, and it has become very simple to take up these essays and study our new drugs. So it has now become very 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 simple, because there are a lot of huge number of literature which is available. But again there are certain diseases where we still do not have good tests. So so identifying a biopsy becomes very very important when we are doing drug discovery, because we should not miss out certain points. So choosing the right biopsy or test system is crucial for success of drug research program. It should be quick, it should be simple, it should be relevant, because there are huge number of compound which we are going to analyze. So that's why we require these assays should be very much quick. Then human testing, at definitely it is not possible. We cannot do drug testing or human testing at first level, and that's why we have to start with in vitro test, followed by in vivo test, followed by pharmacokinetics, dynamics, and safety studies, and then we should go into the clinical trials. So this is how the procedure which is given in schedule Y, or the procedure which is normally followed for any type of drug discovery. So in vitro tests should be cheaper, they should be easier to carry out, they should be less controversial. So these type of tests should be there. Similarly, in vivo test should be such that we should be able to identify the effects, as well as we should be able to identify the pharmacokinetics in these animals. So in vitro, again going back, in vitro tests do not involve live animal. They use tissue, cells, enzymes. Enzyme inhibitor can be tested on pure enzyme in solution. Receptor agonist can be tested on isolated tissue or cells. Antibacterial study can be done by seeing how much the drug kills the bacteria in culture. So these are simple tests, in vitro tests, which are available. Now a lot of cell lines are also available where we can use them for studying the effect of drugs. Similarly, in vivo test. So in vivo test where we are studying the effect of drug on the animals, that often involves a clinical condition in the animal. So in this case, we are developing the disease condition in the animals, and the disease condition, the disease which is being developed in animal must mimic the clinical conditions. So that is very, very important. How we see the disease in clinics, the similar in a similar pattern, the disease should be induced into the animal. Then only we can call that the model is clinically relevant model, and we can test our test drugs. So the animal is then treated in this when we have developed the disease. So these animal can be treated with the test drugs, and we can see whether there is reduction in this disease condition or not. So these type of studies are available and can be done. The animal also, now we are getting transgenic type of animal where certain genes has been replaced or they have been mutated so that they develop the similar condition as in case of human. For example, one example I will give here is the very common example for hypertension, that if you want to develop hypertension in animal, one of the model given is giving the animal replacing its water by NaCl water. So replacing the water by NaCl water, and that is given to the animal. So when animal drinks any sodium chloride water, that means salted water, for a very long time, say around 28 days, it develops hypertension. So this is a common model which is used for studying the antihypertensive drugs. But if we compare this model with the human conditions, how human develop hypertension, so this type of reason is definitely not there. Human do not develop hypertension because of drinking of sodium chloride solution. So then yes, this model is not clinically relevant, and we should have the better model. And that's why we can see that spontaneous hypertensive rat, so these type of rat, these are transgenic rats, has been developed for the research in hypertension. So these spontaneously hypertensive rats, these are the rats which develop hypertension as they go into their adulthood. So in human also the conditions are similar. As the aging happens, as we go into our 40 50s, then the number of cases of hypertension increases. So this is how the model should be. I mean, it should mimic very much to the clinical conditions. So disadvantage of in vivo testing, can we rely on in vivo testing if we have studied our drug in vivo? Can we rely on only on in vivo testing? The answer is no. The example is thalidomide disaster, which we have seen, where we could see that phocomelia was observed in these infants, newborn infants, and somewhere in 1960s. And so that means when we are doing in vivo testing or any type of efficacy or safety studies, the results, we should not rely 100% on these results. So this example tells that thalidomide is teratogenic in rabbit and human, but it is not teratogenic in mice. So when the study for safety was carried out in mice, teratogenicity studies was carried out in mice, the drug was found to be safe. But when it was given to the human, we could see that there were certain disadvantages, or the conditions, phocomelia type of conditions, which was observed in the newborn. So yes, there are certain disadvantages in vivo testings also, and we should be taking care of, because the mouse or the rat or rabbits or hamsters, they do not mimic human body. So we have to study them properly, and then we have to use them as animal models. Okay, the next is test validity. So sometimes the validity of testing procedure is easy and clear. For example, a very simple example is if you want to test the antibacterial activity of the test drug. So we will just see the zone of inhibition, and we will find that the drug is working if the zone of inhibition is high. Similarly, local anesthetic drugs can be easily studied on isolated nerve where we will see that the action potential are blocked. So these are symbols. I mean, there are certain drugs which can be easily studied by the animal testing, by in vitro testing or in vivo testing. But still, we are suffering. There is a disadvantage, or we are suffering from the problems where, especially for development of central nervous system drugs, let it be antipsychotic, let it be anti-Alzheimer's, let it be antidepressant. So these, because it is very difficult to test the drug in animals when the drug belongs to this category. So because the animal could not express. So this is one of the disadvantages we see in animal testings. So that's why we see that the central nervous system drug discovery also takes huge amount of time. Okay, so a lot of time is required for CNS drug development. So thus validity of the test should be carried out. Always we should carry out the validity of test. Every animal model should be clinically validated. High throughput screening is one which is right now we are using, and this is which also has been used for COVID also. And in this case, high throughput screening is miniaturization and automation of in vitro tests. That means a lot of compounds, they are studied for certain receptors, certain enzymes at one time, and we see whether there is inhibition of enzymes, or where there is inhibition of cell growth, or where there is development of certain color. So these type of signals, they are studied, and then the lead compounds are identified. So HTS help us to screen thousands of compound at one time and to find out the lead compounds from this. So this also can be used for the studies for new drug development. Now going next. So once we have seen the we have chosen the disease target, we have chosen the bioassay, so finding out a lead compound is the next question. A lead compound is a compound which shows the desired pharmacological activity. So out of thousands, you will find that 10 molecules are showing good activity, so these 10 molecules should be studied further. So the lead compound will be the one which is having desired activity, and the level of the activity may not be great, and there may be certain adverse effect also. The lead compound therefore provide the start for drug designing. So it is possible, quite possible, that lead compound we have obtained, but it may be having certain side effect. It may not be pharmacokinetically very strong. That means it may not be getting absorbed. So that's all this property we have to check, and then we will come up with, out of, as I said, that out of this 1,000 molecule, we will get only 10 molecule which will be showing good effects. So there are various ways in which the lead compound might be discovered, and so lead compound can be identified by modifying whatever the compound has come successfully. So if they are failing in certain ways, so we have to identify those and we have to manipulate those. We can make them derivatized, or we can formulate them, and then we can use them further. So now let us understand the different steps involved in drug discovery. So different steps involved in drug discovery are from where we are going to get the drug, and how we are going to go about. So first one is screening of natural products. So from where we will get the drug. So either it is coming from the natural product, or it is coming from the folklore use, already people knows, and that is how the drug has come. I gave some example of medical folklore use. For example, artemisinin, which is anti-malarial, developed as anti-malarial. The China has obtained the patent also for this, and it was used initially for malaria long back in their system. So medical folklore also helps. Similarly, we have got some from medical folklore, we have also got some drugs like digitalis, also digitalis alkaloids also. Then screening of synthetic compound libraries. Lot of synthetic compounds are available. Lot many the libraries are available, and we can screen them. We can go for high throughput screening, or we can go for certain receptor ligand binding studies, and we can identify them. Then existing drug, me too drugs, this is called as drug repurposing. Drug repurposing, recently we have seen in COVID also. That means a drug is already been used in other condition, and then it is used for the newer ailments. For example, tetracyclines, which are used for the diseases, the infection which is coming from zoonotic infection, which is coming from animals. So that's why tetracycline, that is doxycycline, was also used for COVID. So this is called as drug repurposing or Me Too drugs. So that means the existing drugs are checked for newer ailments. Then starting from natural ligand or modulator, natural ligands or natural substrate for enzyme product. So these natural ligand can be identified, and then from there we can study. For example, here I would like to give the example of the opioids. So we already have the receptors for opioids in our central nervous system, and further these opioid analogs has been developed. So the receptors are already there, and we have to identify the ligands, and then ligand for those receptor, and these ligands can be identified as drug. Or we also can identify, we have the enzymes in our body, and we can identify the substrate, and that can be used as a drug. So this type of the steps can be taken for drug discovery. Combinatorial synthesis, computer aided drug designing, they are coming up as huge, newer way of discovering the drugs, where we can identify the receptor, and then we can dock our molecule on the receptor and find out serendipity and prepare. So this is one, serendipity always works, and we have seen that serendipity also has previously it has given so many compounds. Penicillin is one of them. So by chance discovery, also the drug comes into the market. Then computerized searching of structural databases, designing lead compound by NMR. So these are all the newer things which are coming up. Screening of natural compounds. So this is first one, screening of natural compounds. So we have already seen that the natural, from the nature we have received so many compounds. Example is vincristine, vinblastine, and they the compound which is responsible for that activity is called as active principle, and they are always the secondary metabolite with quite complex structure, and but they can be considered as novel compound for drug discovery. uh, Disadvantage of getting the compound from natural product is that the extraction is very difficult, it is very costly affair, inefficient process. We may not get sufficient amount of extract. There is change in the, the plant also. If we change the geographical area, then also there is changes in the extract content. So these are certain disadvantages which we see when we are taking the molecules from natural origin. To avoid that, we go for synthesis in the lab, but synthesis is also sometimes difficult because these molecules are very much complex. So these are certain disadvantages which is associated with natural product. So natural product can be obtained from plant kingdom. We have already identified so many molecules from plant kingdom, such as morphine, cocaine, digitalis, quinine, tuberculin, nicotine, muscarine, paclitaxel, and they are used. They all are useful as the drugs, and plant always will continue to remain a promising source of new drugs. Lot of still we are finding many drugs are coming up in a big way from plants. The microbial world, so we know that from microbes also we get lot many drugs like penicillin, cephalosporin, tetracycline, aminoglycoside, chloramphenicol, rifamycins. All these have been obtained from microbial world. Then marine world, from the marine also we are getting many molecules like curacin A, and is obtained, they are obtained from coral, sponges, fish, marine microorganisms. So many they are, and the very interesting thing is they have anti-inflammatory, antiviral, anti-cancer activity. For example, curacin A, which is obtained from marine cyanobacterium, it has anti-tumor type of activity. So marine world also become a source of drugs. Other than that, we have also animal sources as drug. So antibiotic peptides were extracted from the skin of African clawed frog. Epibetidine, which is a potent analgesic, is also obtained from the frog. So animal also become the source of drugs. Venoms and toxin, we get venoms and toxin from animal, plants, snakes, spiders, scorpion, insect, microorganism. So we know that this teprotide, which is obtained from viper, is the lead compound for antihypertensive drug, which is captopril and cilazapril. So yes, we get from venom also the drug is obtained. Then second is medical folklore. So in medical folklore, the berries, leaves, roots which are used by local healers, they are used as medicine, and so many of them we have obtained from this folklore use. These are example: quinine, reserpine, atropine, digitalis, cocaine. So when we get it from the from the people who are using it in other village places, or which has been known traditionally, so they also can work as new drug, can be used as information for new drug discovery. Then screening of synthetic compounds libraries, already I said that thousands of compounds have already been synthesized, and majority of these compounds are never been used or they are not in the market. So these compounds are available in the libraries, are they can be used for testing. And pharmaceutical companies, they have this type of library and they keep on testing these compound with the targets. Existing drug, me too drug, already I said that many companies they use established drug from other competitors as lead compound in order to design a new drug. So off patent, when the molecule becomes, they can modify and they can develop the new type of drugs. Other than that, drug repurposing is also coming in a big way. Then enhancing a side effect is again one area that, for example, a side effect of a drug can be considered as effect. So this is how the drug can be developed. For example, sulfonamide, which came up as antibacterial, they had a side effect of hypoglycemia. So these sulfonamide were further studied and they were developed as sulfonylureas as anti-diabetic drugs. So sulfonylurea example tolbutamide. So these type of side effect can be converted into pharmacological effect. One more example is minoxidil, which is a vasodilator, which initially came for hypertension, but now it is mostly reserved for hair growth. So enhancing a side effect is one of the we can use it as new drug discovery. Starting from natural ligands or modulator, natural ligands already which are uh, there in our body. So natural ligands of a target has been sometimes used as a lead compound. For example, adrenaline, noradrenaline is already there, neurotransmitter which are there in our body, and they are used for development of beta agonists such as salbutamol, dobutamine. So salbutamol, dobutamine, they have structure similarity with that of adrenaline, noradrenaline. Similarly, antagonists also can be developed. So already these molecules are in our body. We can understand their pharmacology and then we can develop them. Either we can develop the agonist or antagonist, depending, and we can identify their structure, and on that basis we can develop agonist or antagonist. So okay. Then once this all is done, where we have identified from the drug is from where the drug is going to come. So the last part remains is last few parts they remain is isolation and purification. So if the lead compound is present in a mixture of the compound, it has to be isolated and purified. The isolation and purification, it depends upon structure, stability, quality. So either we are getting it from natural origin or we are getting the drug from synthetic origin. Finally it has to be in the pure form. We cannot give mixture as a drug. So it has to be in the pure form. One example I'll give you is that Fleming identified recognized penicillin. He by serendipity he identified penicillin. So he suffered from lot of problems with the isolation, because he was unable to isolate the pure penicillin, and whenever he was trying to do it, the drug was getting destroyed. So purification and isolation of penicillin were possible until development of new experimental procedures such as freeze drying and chromatography. So that was again one of the drawback of Fleming's experiment. So yes, we require the drug which should be in the pure form. It should have a structure, it should have stability, and in the sufficient quantity. Structure determination, we have the techniques available: X-ray crystallography, NMR spectroscopy, mass, and IR are there important in structure determination. Nowadays, we see that we have, these labs are available everywhere and we can do it. Now let us go next part. Once you have identified the molecules, either it is coming from any of the origin, so these molecule will we will go for their development as a drug. And but before developing it as a drug, we have to study this molecule in preclinical setup and in clinical setup, that is preclinical trials and clinical trials. So traditional medicines, clinical trials of the herbal drug, here I would like I'm just putting because the uh, the audience right now who are sitting here, they have the background of Ayurvedic medicine as well as herbal medicines. So traditional medicines, they are governed by Drug and Cosmetic Act 1940 and rules thereof 1945. In 1959, Government of India amended the Drug and Cosmetic Act to include the drugs which are derived from traditional Indian medicine. The procedure laid down by the Office of DCGI for allopathic drugs should be followed for all the traditional and herbal products. So this is one thing which we have to here see, that for traditional herbal product drug development also we have to follow the guideline which are given for allopathic medicines. So same way we have to develop the our molecules also. So drug development approval process, let us see. So we have to initiate our drug development process with the help of preclinical studies. And in preclinical studies, that means we will be studying these drugs which we have identified or isolated from different sources. We will be studying them in animals, which will be a long-term animal toxicity study. Then also during that time when we are doing preclinical study, we also can go for formulation development, manufacturing control, packaging designing, all can be done together. We also have to apply for IND, investigational new drug application, and then we can go for clinical trials such as phase 0, phase 1, phase 2, and phase 3, and phase 4 type of clinical trial. This diagram shows how the overall procedure is. So preclinical research and development is the first stage for developing the drug now, and which takes around 6.5 years. So around 6 years, 6 to 7 years it takes. Then is the clinical research, clinical studies. So in clinical study we have phase 1, phase 2, phase 3, which takes around 7 years. Once we have done clinical studies, we have to go for we can go for NDA application, which the documentation is itself is taking 1 to 2 years. So that also we have to consider. And then after that we can go for post marketing surveillance, and which will also include phase 4 trials. And so this is how the we see that 12 to 15 years of time is required for developing any drug. So this picture summarizes that. Now let us understand the phases of clinical trial. So phases of clinical trial are phase 0, 1, 2, 3, and 4. 0 is human microdosing studies. 1 is first in human, that is done in volunteers. Phase 2 is in the patient. Phase 3 is in the large scale multicentric trials. And phase 4 is post registration monitoring trials. Preclinical study happens before phase 0, and this involves we are testing the molecules in the animals, and they are called as efficacy studies and toxicity studies. Efficacy we have already studied, in vitro studies and in vivo studies, whereas toxicity studies we have to carry out acute toxicity studies, or acute or chronic type of toxicity studies, oral toxicity studies, if it is intended to be given orally. The guidelines are already been mentioned by OECD guidelines, and there are different guidelines which are mentioned for different type of studies. And for acute oral toxicity, we have AOT 420, 423, 425 type of guidelines already available, which we have to follow when we are doing these type of studies. The details are given. I will not go much in detail for toxicity studies here. Other than that, we are also doing special toxicity studies such as effect on reproductive performance, teratogenicity, carcinogenicity, mutagenicity, and local toxicity also, if the drug is intended to be applied dermally, or if it is intended to be instilled into the eye or into the vagina. So this type of specific toxicity study also has to be carried out as per the guideline. Now, these are the toxicity studies, subacute or subchronic toxicity studies, where the animal is dosed for 2 weeks of daily drug administration, and at 3 or more doses level, and the animals are studied for different type of effects for morbidity and mortality. Then chronic studies, where we are again dosing the animal for 90 to 180 days, and we are demonstrating the safety of the molecule. If the drug is to be used for chronic human illness, animal studies 1 year or longer must be taken in support of human use. So it is possible that because most of these studies are 90 to 180 days, but in case if the drug is required to be used in human for more than 1 year, then 90 to 180 days studies, which are called as chronic studies, there we have to increase the number of days of treatment in the animal also. Carcinogenicity studies also we are taking into account when we are doing the toxicity in animals. So the usually the component of chronic testing and is undertaken when compound has sufficient promise as a drug to enter the human clinical trial. Carcinogenicity must be done if the human clinical trials are required to be carried out. Carcinogenicity studies are long-term studies, 18 to 24 months, with surviving animal killed and studied at defined week and during the test period, and data on the causes of animal death, tumor incidences if it is seen, type and site, and necropsy studies are collected and evaluated. Reproduction studies are also undertaken to reveal any effect of the new drug on mammalian reproduction. So these studies are fertility and mating behavior and early embryonic study, prenatal study, postnatal development. So these studies already the guidelines are they are giving the effect on reproduction. So this type of study should be always carried out. In this study, the maternal parent, fetus, neonate, weaning offspring are evaluated for any type of abnormality, growth, and development. Embryotoxicity study, only second mammalian species traditionally has been required. Rabbit is a preferred choice for practical and extensive background knowledge accumulated on this species. So that also is required. Genotoxicity, mutagenicity, if any changes are happening in the DNA or that is damage to the DNA or any type of gene mutation is happening. So this type of study also is required to be done for the new drug discovery, which is called as genotoxicity or mutagenicity. And here we are using Salmonella typhimurium for detecting the toxicity. Other than that, all these safety trials, which is required compulsory to be done for new drugs, we are also required to carry out pharmacokinetic studies. In pharmacokinetic studies, the normally the preferred animals are rat and dogs and monkeys. So higher animals are mostly used, dogs and monkeys are mostly used. So ADME, complete absorption, distribution, metabolism, excretion pattern of the new drug is studied in these animals. Bioavailability is determined for the new drug. What is the rate and extent of absorption of the drug in the blood? So that is determined. What is the T half of the drug? How much time is required for its excretion? So this also has been studied. Pharmacological studies also involves therapeutic index, ED50, LD50 estimation. So all this whatever study which we are doing, toxicity as well as kinetic, will help us designing the studies in human. So what I have discussed right now is toxicity studies, and the second is pharmacokinetic studies, which are very important for preclinical studies. So other than that, when we have done toxicity testing, PKPD, the other is efficacy trials, which are called as pharmacological studies. So efficacy trials are also important, where we will be developing the one type of disease in the animal, and then we will study the effect of our test drug. For example, if it is diabetes, so we will develop diabetes in the animal, and then we will study the effect of our drug in diabetic animals. So overall, if we see preclinical evaluation, it takes a lot of time, almost 1.5 to 3 years, almost 1.5 to 3 years, and out of 10,000 molecule, we find that only 10 molecules they passes the preclinical evaluation. Lot of failure rate is seen in preclinical evaluation. The possibility of failure is that it may be toxic, so in if a safety trial they are removed out if they are found to be toxic. And second is when their PKPD is not so good, so if their kinetic is not so good, if they are not getting a job, in vitro they are showing the effect, but when we have studied them in their when we have studied them in the animals, so in vivo studies, then we are finding that the drug is not getting a job. So these type of failures will lead to only 10 molecules out of 10,000 molecules. Simultaneously, we also have to carry out for these newer drugs which will be going into clinical trial, the early formulation study, because these are the drug which are given to the animals are in the form of solutions. But when we are trying to give it into, when we will be giving it to the human, then the drug will has to be in the form of formulation. We cannot give the extract, we cannot give the powder, we cannot give the powder dissolved in the solvent, we cannot do that. So it has to be converted into formulation. So early formulation studies are simultaneously carried out. As a promising compound is characterized for biological activity, it is also evaluated with regard to chemical physical property that have bearing on its ultimate and successful formulation into stable and effective pharmaceutical product. So this is the area of responsibility of pharmaceutical scientists, formulation scientists who are trained in pharmaceutics, and they will develop this molecule into the either into the syrup form, or into the into injectable form, or it will develop into tablet form, capsule form, or into the aerosol form. So it depends upon how the molecule is working. Each drug substance has intrinsic chemical and physical characteristic that must be considered before the development. And drug solubility, partition coefficient, dissolution rate, physical form, stability, all these are important characteristic for development of formulation. So if the drug is not, if the drug is physically unstable, then it will be very difficult that whatever the effect we are getting in the human is because of the drug, because it is physically unstable. It get degrades because of photosensitive drug. So we have to consider all these points during pre-formulation studies. Initial product formulation and clinical trial materials. So in this case now, we have to prepare for phase 1 and phase 2 for clinical trial. Phase 1 studies for orally administered drug capsules are normally employed that consist of active ingredient with or without pharmaceutical excipients. So the very common way of giving the drug is in the form of capsule. Fill the powder in the capsule and administer it. We have to take the approval before initiating the clinical trial from human ethics committee, and human ethics committee is responsible for seeing that our study is done in a very ethical manner, in a very scientific manner. So informed consent has to be taken from the patient in verbal as well as written manner as per the guidelines, and then we have to start our study. Placebo is normally employed in most of the studies. In certain study only it is not used, but most of the study placebo is employed, which looks similar as drug but do not contain the active ingredient. So let us start with clinical trials. The clinical trial, first one is phase 0 trial, and it is also called as human microdosing studies. So in human microdosing studies or phase 0 trial, the objective is to study the pharmacokinetics, particularly oral bioavailability and half-life of the drug. So we are giving now first time into the human. The drugs are required to be given. So that is why we are using the healthy volunteers. 10 healthy volunteer can be used, and for the first time we are just interested right now into the pharmacokinetics. That means whether if the drug is given in the oral dosage form, in the form of capsule, whether the drug is getting absorbed through the GIT, yes or no. If it is getting absorbed, then only we can proceed with the further clinical trial, otherwise it will be of no use. So this microdosing study is for that purpose, and that is why we are using healthy volunteers, and we are using subtherapeutic effect, a subtherapeutic dose of the drug. Subtherapeutic, why? Because we are using for the first time in human. Quite possible that the drug may show toxicity, though we have tested already the toxicity of this molecule in animal, but in human for the first time we are using. So that's why it may show toxicity, and that's why we have to go for subtherapeutic dose initially. Now phase 1 trials. Now first, once we have obtained the kinetic of the drug in the phase 0 trial, we can go for phase 1 trial, which is first in human trial, again called as, in this case, to check whether animal and human show any differences. The pharmacokinetic variability between individual, effect of route of administration, bioavailability is okay, tolerated dose range, indication of therapeutic effect, and indication of side effects. So these are our objectives. So what we are testing in this first in human trial, phase 1 trial, is that already we have received certain signals from phase 0 trial that this is the dose, subtherapeutic dose, and there is no toxicity. So in this case right now what we are going to study is we are going to use the therapeutic dose, and we will see whether there is any problem associated with the therapeutic dose in terms of side effects. and we will also study the kinetics, pharmacokinetics, and if any variability is there, that also we are going to study. Now, phase 1 trial involves the clinical researcher, then it involves healthy volunteers. Informed consent is taken from healthy volunteers. Medical backup is always maintained. The monitoring team is always there. Single and repeat doses or increased dose level can be done. Formulation can be given here, and safety, it's a safety trial. So here we involve lesser than 100 healthy people, and it is designed to determine that the drug is safe and there is no side effect. So this is the main purpose. Less than 100 people we are using, and we are using the dose which is now therapeutic, and we are finding that the drug is safe in these healthy volunteers and there is no side effect. We are also finding out the pharmacokinetic. Already, as I said, that we will be studying pharmacokinetics. So that means we have administered the drug to the healthy volunteers, and simultaneously we are removing the blood at definite time intervals, and we are studying how is the absorption pattern and how is the excretion pattern, what is the bioavailability, what is the T half of the drug. So all these pharmacokinetic parameters we will be able to obtain. It also, we also can find out the maximum tolerable dose, MTD. So also can be done. Next is phase 2 trials. So once phase 1 trials are over, then phase 2 trials are done, where we are taking 100 patients with the disease now. So now they are the patients and they are with the disorder or disease. Now we have to evaluate the doses needed, detail how and why drug works in the body. So are now are becoming more, we are trying to understand mechanism also. We are trying to understand side effect it causes at that dose in the patient. So that also we are finding out, and the drug must be effective and safe. Now, these phase 2 trials are phase 2A and phase 2B type. So, phase 2A is pilot clinical trial. These are pilot studies which are which are carried out to evaluate the efficacy and safety in selected population of the patient with the disease condition to be treated, diagnosed, or prevented. Objective focus on dose response, type of patient, frequency of dosing, numerous other characteristic of safety and efficacy. So phase 2A trials, whereas phase 2B trials are again safety and efficacy in the patient with the disease condition. These clinical trials are usually, they represent the most rigorous demonstration of medicine's efficacy. Sometimes they are called as pivotal trials also. So phase 2 trials, we are studying in the patient, and the phase 2A with where um, we are they are not very strict, but phase 2B trials are most rigorous. So we will identify, we will, let us see in this case now. So in this phase 2 trials, so more number of patients are taken. Phase 2 where we are taking the patient, we are endpoint is decided now. What is the endpoint? So for example, if it is diabetes, the blood glucose has to be lowered down, so that is the endpoint. So endpoint is decided, maximum monitoring is done. Patients, often patient where the other treatment failed, so there also we are taking those patients. Then again, as I said, that there is the phase 2A and phase 2B. So phase 2A is where the rigorous treatment is not given, and 200 patients, and it is single blinded type, whereas phase 2B is a double blind, where the patient as well as the physician does not know what drug is given to them. So this is how the phase 2A and phase 2B trials is done. How, what we are getting, we are, what objective is uh, answered in this trial. So indication for use is identified. So what, for what purpose the drug is, what is the so because we are doing in the patient, so we are identifying that, this is the use of the drug, indication is identified. Type of the patient, whether the severe patient can be also be treated, or whether it is mild to moderate disease can be treated, so that also we identify. We also identify the dose range of the drug. We also identify the schedule and increment of the dose drug. Pharmacokinetic studies also can be done in ill patient. How the drug behave in ill patient can also be studied. The nature of side effect and severity can also be identified in these patients. And effects in special group also starts coming up. Now going to next trial, which is phase 3 trial. So phase 3 trial is larger group of patients, around 2000 to 3000 patients are taken, and it is multi-centric type of trial. Lot of centers are where these trials keeps on going at simultaneously, and we are comparing the drug with the existing drug. Now the test drug is now compared with the existing drug, and because huge number of patients are taken, so already they must be taking some other drug also, so that type of interaction also can be studied here. It also provides statistics on adverse reaction. So phase 3 trials, first is phase 3A trials. The trials are conducted after efficacy of medicine is demonstrated, but prior to regulatory submission of new drug application or other dossier. So all the trials which are carried out before submitting the application, that is new drug application, two application is made to DCGI in India. One is IND, that is to carry out the clinical trial, initiate the clinical trial, and second is NDA. NDA is done after the phase 3A trial. NDA is done. So this phase 3A trial is done before submission of NDA. These clinical trials are conducted in patient population for which the medicine is eventually intended. Phase 3A clinical trial generate additional data on both safety and efficacy in large number of patients. So here we are getting large number of patients. We are also identifying the group, special groups, as I have mentioned previously also. That means renal failure patients or some other condition patient. So in these patient also we identify how the drug is behaving. And also we also, because they are already taking some other drugs, so drug interaction also comes out. Many adverse effect also comes out. So all these points can be identified now in 3A because the patients are huge. 3B is clinical trial conducted after regulatory submission of NDA or other dossier, but prior to approval. So this trial supplement the earlier trial and complete the earlier trial, and may be directed towards new type of trials, that is quality of life, marketing, or phase 4 evaluation. This is the period between submission and approval of regulatory dossier for marketing authorization. So 3B is just a continuation of the previous trials, only they happen after submission of NDA. So NDA or other dossier. So in this case, overall phase 3 trial include thousands of patients. It depends upon prevalence of the disease. Multicentric trial it is randomized type of trial. It is double blind. Double blind randomized means anyone can get placebo or the drug. Double blind is physician doesn't know what drug is given to whom, whether the patient is administered placebo or whether the patient is administered drug. Even the patient does not know. So it's a double blind type of trial. More certain data for objectives of phase 2 studies. Interaction between drugs start to become measurable. Subgroups start to be established. Special feature and problems are shown. Once the three beat after 3A trials, they can apply for NDA, and so sponsor can file the application for NDA. Complete monograph insert can be now done, and once the NDA is done, submission is done, FDA has reviewed, and pre-approval inspection, plant inspection is carried out, and FDA can approve it, or it may also disapprove it. Anything is possible. But once the approval is given for the new drug, the drug is comes into the market, and as new drug, and phase 4 trial keeps on happening after approval of marketing. So post, it is also called as post licensing phase. There is no fixed duration for how much this has to be done, but it is the manufacturer has to submit the report every 6 months for 2 years, and then annually for next 2 years. So this is to identify the rare type of side effect or congenital effect if it happens somewhere in the population. So phase 4 trial is continuous trial, and it is for identification of certain type of side effects that may be seen in the population. So phase 4 trial studies or trial conducted after a medicine is marketed. It provides additional detail about medicine efficacy and safety, different formulation, doses, duration of treatment, medicine interaction, medicine comparison is evaluated, new age group, races, other type of patients are studied, detection, definition of previously unknown, inadequate adverse effect can be now quantified, and risk factor can also be identified. So these are all the objective of phase 4 trial, which is happening post marketing. The very common side effects which are seen are they are like, drowsiness, they are hives, diarrhea, or dizziness, headache, rash. So these are the common side effects which are seen with the drugs, which can be also reported in phase 4 trial. So overall, the journey is long for new drug discovery, and it starts with the lab or synthesis or from natural origin. It is tested in animals in preclinical trials. Then it goes into the healthy volunteers, into the microdosing trial or phase 0 trial, and followed by phase 1 trial, and then in the patient, which is phase 2 trial, and phase large number of patients in a multicentral trial, phase 3 trial, and followed by it comes into the population once it is been approved by FDA. The even after phase 4 trial, lot of drug has been withdrawn from the market because of many reasons. The very important one which I would like to tell you is this is the list which is given here, but rosiglitazone, which came as a oral hypoglycemic agent, but it caused increased risk of heart attack and death, and that's why it was withdrawn. Sibutramine, which was anorexiant, and which increased the cardiovascular risk, again was withdrawn. So many drugs, they come into the market, they are studied for a long time, but then they are because of the side effect, they are withdrawn from the market. Rofecoxib is one of such drug, which came as analgesic anti-inflammatory, but it was withdrawn because of risk of myocardial infarction. So other than that, also these were the known drugs, but other than that also there are so many drugs which comes and then which goes off the market. There are many drugs which has been approved recently, and mainly right now the drug development is happening in COVID because COVID pandemic is huge. And Spikevax, which was developed by Moderna, then Liquio, which was developed by Novartis for treatment of hypercholesteremia, then Aimovig, which is developed by Amgen for migraine, Illumia, which is developed by Sun Pharmaceutical for psoriasis. So these are certain diseases. See, we can still see development: psoriasis, migraine, and hypercholesteremia. But I again feel that the better treatment is hidden in Ayurveda and not in allopathic system of medicine, for especially for diseases like psoriasis, for migraine type of disease, for hypercholesteremia, for cardiac associated disease. So we have to think in this direction and develop drug for them. So with all these slides, I thank all the audience for their patience listening, and I'll be happy to answer also. So thanks a lot.
Thank you very much, madam, for this elaborative session. It was very nice and good explanatory. If anybody is having question, please ask. You can ask in chat box. We'll wait for 2 minutes if anybody is having question. Otherwise, we'll stop there.
Chinmay sir, hopefully that I was just thinking from the point of view that how we can include Ayurvedic medicine in the in the course of drug discovery, how we can develop them as newer drugs, because lot of very less, very meager studies is been done in Ayurvedic medicine in India. Please refer to certain, I would also like to take name of Dr. Bhushan Patwardhan from SPPU, and his publications are available where he is talking about the drug discovery which is happening at a very slow pace in Ayurveda, because we are, it is not coming as a mainstream drug. So that is where we have to think, why we are lagging and why it is happening. I mean, when we compare with, as I said, I just compared it with Chinese medicine, traditional Chinese medicine. So traditional Chinese medicine, almost the data shows that many of them are in the way of getting approval by US FDA. US FDA, many of them, two of them already got approved, and many of them are on the way. But for Ayurvedic medicine, we still do not have a single drug which has been approved by US FDA. So there, I think there is a where we can work together, and we can think of developing it as a newer drug. And we all of us, we know that Ayurveda has huge potential, and we have seen that in the case of COVID also, during COVID also, and it has huge potential. We have to just see that, how, what is the mechanism of action, how we can make it as a drug, Ayurvedic drug as a main drug, allopathic drug type of thing, something like that.
Yes, yes, sure. Most of the formulations in Ayurveda are classical one, and but those are used clinically, but less research has been done on it. But we'll
Yes,
surely think on it.
yes, yes, yes,
Yes, I think no question is there. We'll end up with this session.
yes, we can leave.
Yeah, sure,
Thank you, sir.
sure, sure.
Thank you.
Thank you very much.